Thank you for the introduction. So indeed, I'll talk about the evolution of sex chromosome and how fungi can contribute to understand the evolution of sex chromosome, because, so as you know, many animals, in particular mammals, have a very differentiated sex chromosome. Like here you can see the X and the Y sex chromosomes in mammals. But in fungi, like you can see here, mating between two gametes in the mycobottium fungi. So they only mate if they are of opposite mating types. So we call it them A1 and A2 here. So they mate and go hyphae. But you can see there are not many different traits between these different mating types. It's not like males or females. And despite that, despite the absence of different sexes, we find similar pattern between mating type chromosome and sex chromosome. So we try to understand why there would be a shared pattern and whether we can have a general explanation, general theory that can hold both with for sex chromosome and mating type chromosomes. So I'll talk about this shared pattern of stepwise extension of recombination suppression across eukaryotes, so in animals, but also in plants and in fungi, and also around supergenes in butterflies and ants. So it looks like once there has been a first step of recombination suppression, it can extend with time. Stepwise, so the initial recombination suppression become bigger and bigger in several steps. And so this was known in sex chromosome in animals and plants, but we found the same pattern in fungi and the same pattern in other supergenes. So we thought we need a general explanation for all that. And so we try to develop a new model based on the sheltering of deleterious mutation. So I will present that too, and how fungi could also help to validate the model prediction. And then we also know that after recombination suppression evolves, there can be accumulation of the deleterious mutation because selection is less efficient. And so we have also studied the pattern of deleterious mutation accumulation after recombination suppression. So we studied both deleterious mutation as a possible cause for recombination suppression and also as a consequence of recombination suppression afterwards. So first about the shared pattern of stepwise extension of recombination suppression. So as you probably all know, there is often restriction as for mating across eukaryotes. So not all gametes are compatible with all other gametes. There is specific loci in genome controlling mating compatibility. And these loci often show recombination suppression, and that's true on sex chromosome, but also incompatibility loci, like self-incompatibility loci in plants, but also in fungi, the mating type loci in fungi. And so we understand why there is a recombination suppression around this gene, because they function like key and lock key and lock model. So to remain incompatible with the alternative form, you need the suppressed recombination between several genes that function like the key and the lock of the other, the alternative alleles. But what is more puzzling is why are sex chromosomes sometimes so differentiated? So if you need recombination suppression just at the sex-determining gene, so we understand why, but why has the recombination suppression been stopped across the whole X and Y chromosome? And this is still debated. So one part of the answer is that recombination suppression extended progressively with time. So initially there was recombination suppression just around the sex-determining genes, but it extended with time. And how do we know that? And we know that for sure. It's when we plot the differentiation gene per gene between the X and the Y chromosome, for instance. And so this is a proxy for time since recombination suppression, because as soon as recombination is suppressed, the two alleles accumulate different mutations and they will become more and more differentiated with time. So if we plot the divergence between the X and the Y allele, we have a proxy for the time since recombination suppression. And if you plot this pair gene divergence between the X and the Y alleles along the X chromosome gene order, you can see in mammals and in some birds, in some plants, some what we call evolutionary strata of differentiation. So you have a discrete strata of different level of divergence. And this indicates that recombination suppression occurred first in here the central region, and then extended with time in several steps, because you have strata with decreasing divergence when you go farther from the sex-determining gene. So this is a pattern. This is known for sure. There has been stepwise extension of recombination suppression and in multiple lineages, in plants, in animals. But the question is why? So why does recombination suppression extend with time? And this is still not very well understood. And so the main hypothesis for a long time has been sexual antagonism. And the idea is indeed appealing and quite simple and straightforward. And the idea is that you have many traits that are different between males and females, right? You can see the quorum in lions, the colors in birds, the flower size in Silene latifolia. And so you have multiple genes with alleles that are beneficial in males, but not in females, and conversely, some genes that have alleles that are beneficial in females, but not in males. And so it makes sense that selection should favor their linkage to the sex-determinant gene. And so these green genes will be the gene with sexual antagonism, so with alleles beneficial in only one sex. And so if by selection you link this gene to the sex-determinant gene, you will have the male beneficial alleles only on the Y chromosome, and this should be beneficial. So this idea makes sense, and the model, theoretical model works, but actually, despite decades of research, there have been very little evidence that indeed this model apply in nature and cause the evolutionary strata. In particular, most of the sexually antagonistic genes actually are on autosomes, and they are hormonally regulated. And actually, we found also evolutionary strata on other around other regions with our combination. So on the supergenes that control the color of the butterfly wings, for instance, or the social structure in ants. So you have a first recombination suppression in a small region, and that extends with time progressively. And here there is no different sexes, right? But you could still say that there are different morphs, and they could also incorporate different genes that are beneficial in one social structure and not the other, and so on. There could be some other kind of antagonism, even if not sexual. So we thought maybe we could study organisms that do not show any sexual antagonism or other type of antagonism. And if we find strata in this organism, this means that other mechanisms can work and generate this evolutionary strata, and that this could also apply on sex chromosome. So we study this microbiome fungi. So they are pathogenic fungi. You can see that they produce the spore in the anthers of the plant. So they castrate the plants by preventing them to produce pollen or seeds. And so they produce the spores instead of the pollen. And then the spores are dispersed by the pollinators that usually disperse the pollen. And what is interesting is that we call them anther-smut fungi. They can parasitize many plants of the Caryophyllaceae family, and they are like basically one species per plant species, so multiple species of fungi. So the wide range of like hundreds of species, specialists each on their host plant. And you can see here two gametes of the fungus mating so they can mate only if they have different mating types, what we call A1 and A2. But you can see that there are not many traits different between A1 and A2. They're just haploid cells, and so there cannot be much of antagonism between mating types. And you can see here actually a drawing by Charles Darwin in a letter he wrote to Lydia Baker. He became interested in this disease, mostly not in the disease itself, but in the change of phenotype it caused to the plants, because in Silene latifolia, that is dioecious, so with female and male plants. So even the female plants actually grew anthers full of spores. So you can see here a drawing by Darwin where he showed that the female structure, when they are infected by the fungus, the ovary abort and they grow anthers full of spores. And so you have here a spore of the fungus. The spores that are in the flower are diploid, and then they germinate, they undergo meiosis, and they produce 4 haploid meiosis. And actually, so 4 haploid products of meiosis, 2 of A1 motic type and 2 of A2 mating type. And actually, they mate within the tetrad without any haploid phase, actually. So there is no haploid phase where they are of different mating types. So there's really no room for antagonistic selection between mating types. There is no room for any selection to be a better A1 or better A2, where you could select for linking genes with antagonistic effect. So that's an ideal model. If we find evolutionary strata here, it means that other mechanisms that antagonistic selection can generate this evolutionary strata, and that could also apply to sex chromosome. So a bit of background on the mating type in fungi. So these fungi are basidiomycete fungi, like the mushroom and the rust and the other smuts, like the Ustilago smut that caused the delicacy that we eat, the huitlacoche in Mexico. So the mating type in these basidiomycete fungi are controlled by 2 set of loci that are themselves actually supergenes, so several genes linked together by suppressed recombination. And in most basidiomycete, these 2 sets of genes are on different chromosomes. So there are 2 loci controlling mating type. So one locus we call the Pierre locus, with pheromone and pheromone receptor gene that control the possibility of fusing, gamete fusion. And the HD locus with homeodomain genes, that 2 homeodomain genes linked together that control the post-mating growth of the hyphae growth. And so for making a viable hyphae, you have to have gamete fusion between gametes that bear different alleles at the 2 loci. So you have to carry different loci at the Pierre locus and different alleles at the HD locus. And so the fusion occurs so at the haploid stage. So actually mating types in fungi do not prevent selfing, because so you have haploid cell of different mating type that mate. So all diploid individual will be heterozygous for the 2 mating type loci. So you can have selfing, and actually the Mycobacterium fungi do self a lot. So they mate within the tetrad just after the meiosis. So they do self a lot. And actually, in the selfing fungi, we often observe that the Pierre and HD loci are linked together by suppressed recombination. And so we think this is beneficial under selfing, because so if the 2 loci are unlinked, you produce 4 mating type after meiosis, while if they are linked, you have only 2 mating type after meiosis. And so for a given individual, when the HD and Pierre loci are linked together, one particular gamete will be compatible with half of the other gametes, while if the loci are unlinked, they will be only compatible to 1/4 of the other gametes. So we understand why there could be suppressed recombination between the HD and Pierre loci, but then the question is whether it extends beyond the mating type loci in evolutionary strata. So we sequenced the genome of many Mycobacterium fungi. So you can see here the picture of the disease plant. And so with 1 Mycobacterium species per disease plant. And you have here the phylogeny of the Mycobacterium fungi. And first, we found something very surprising. It is that actually the linkage of the HD and PR loci occurred not just once at the basis of the mycobacterium clade, but repeatedly and several times independently. So each time you have a red arrow here, it indicates an independent events of HD and PR loci. And actually, so we know that by several kind of evidence. And the first evidence is that actually, so the ancestral state is that you have a PR and HD chromosome separated. And when they are linked together, we infer the chromosomal rearrangement. So for instance, for all these species with the one here, you have the whole PR chromosome that became linked to one arm of the HD chromosome. But in other species, it's another type of rearrangement that link together the HD and PR locus. So for example, in the case 2, you have the PR locus, PR chromosome became linked to the arm of the HD chromosome, but in the other direction. So it's another event, obviously, because it's a different rearrangement. In other species, you had the whole PR and HD chromosome that became linked together. In other species, it's only one arm of the PR chromosome and one arm of the HD chromosome. So there have been several cases, several types of rearrangement showing independent event of rearrangement and linkage of the HD and PR loci. And in some other cases, actually in the C case here, you have the HD and PR loci stayed on different chromosomes, but they each became linked to their centromeres, which means they both segregate at the first meiotic division, which gives actually exactly the same odds of compatibility between the gametes within a tetrad. Half of the gametes are compatible with the other half. So you have had, under the same selective pressure, repeated evolution of chromosomal rearrangement in different ways, but each time linking the HD and PR loci, or another solution, another evolutionary solution, linking the loci to their centromeres. And you have had several, like at least 11 events, independent events of recombination suppression, linking the HD and PR loci together or to their centromeres. So that was a first interesting results. So the different rearrangement were a first indication of different events, but also the transspecific polymorphism showed independent recombination suppression events across the phylogeny. So the idea is that we look at the gene genealogy as genes that are between the HD and PR loci and the mating type chromosome. And so as soon as you have suppressed recombination here, you have accumulation of different mutation in the A1 and A2 mating type. And so the alleles became differentiated. And so like I showed previously on the X and the Y chromosome, the differentiation between the alleles give you a proxy for the time since recombination suppression. And if recombination suppression occurred before, after speciation, so the gene genealogy will show like this, with the A1 and A2 alleles of each species branching together. But if the recombination suppression occurred before speciation event, actually all the A1 allele will branch together and all the A2 allele will branch together, regardless of the species, right? Because the A1 and A2 alleles will begin to accumulate different mutation even before the speciation, and they cannot recombine, so they will stay different. So the point here, the node, is the gene genealogy really indicates when recombination suppression occurred. And this was another indication, another strong indication that recombination suppression occurred several times independently across the phylogeny. And so we looked whether there were evolutionary strata, whether there were stepwise extension of recombination suppression beyond the mating type loci. So we did like is typically done in the X and Y chromosome. So plotting the divergence between the alleles associated with the A1 and A2 alleles along the mating type chromosome. So when there is recombination, you have zero divergence because it's highly selfing, it's highly homozygous. So we have no divergence between the A1 and A2 genomes. So it's very convenient because you can really say when there is recombination because the plot is flat. And so when there is recombination suppression, you have divergence between the A1 and A2 associated alleles. And you can see here, it doesn't seem to be any evolutionary strata, right? It's just a chaos of different divergence levels. But the difficulty here is that actually it's not like in the X and the Y system, where the X chromosome still recombine, so keep the ancestral gene order, so you can trace back the history of evolutionary strata. Here, the two mating type chromosomes, none of the mating type chromosomes ever recombine. They are both always heterozygous. So they both undergo rearrangement. And indeed, when you see here a plot showing the synteny between two autosomes in one diploid individual, so they are perfectly synthenic because they are recombining. And you can see here the plot, so linking the genes of the two mating type chromosomes. And here it's just chaos of rearrangement, right? Except at the two edge of the mating type chromosome, where the pseudo-autosomal region remain collinear and recombining. Otherwise, both chromosomes are just completely rearranged. So, of course, we cannot see any evolutionary strata plotting the divergence along the current gene order. You need the ancestral gene order to reconstruct the history of the evolutionary strata if there have been first recombination suppression around the mating type gene and then away from the mating type gene, but you need the ancestral gene order to see that, right? So we looked for the ancestral gene order by sequencing outgroups that had still recombining mating type chromosome. So we looked at these two species that are mating type. So this one has mating type chromosome that are still recombining on different chromosomes. So this Mycobacterium lageremi. So the mating type loci are linked to the centromere, but so very recently. And so here, when you compare the synteny of the mating type chromosome, so the PR chromosome, the HD chromosome of these species against these other species that are very distant, right? They have remained synthenic, these two ones. So because they are so distant in the phylogeny, if they have the same gene order on the mating type chromosome, it means that they have kept the ancestral gene order, right? They cannot have by chance the same gene order that was not shared by the ancestor. So we consider that was a proxy for the ancestral gene order. So that's just a small rearrangement here. So then we plotted the divergence between the A1 and A2 alleles, but this time along this reconstructed ancestral gene order. And here, now we can see footprints of evolutionary strata. So you can see high divergence for the HD genes and PR genes. And you can see that around each of the HD and PR genes, there are clusters of genes with high divergence, higher than between the two loci. And then when we go farther from the PR gene at right, there are different strata with different levels of differentiation. And so we separated this evolutionary strata with different color. So not only based on this differentiation plot, but actually we defined them by looking which species had suppressed recombination in this region across the phylogeny. So you can see here at right that all the Mycobacterium species had recombination suppression around the HD and PR genes, so that we call the purple and blue strata. So this bump of divergence around the HD gene in blue and around the PR gene in purple, all the species had them. Even the species that had still recombining a mating type chromosome, that they still had recombination suppression around each of the PR and HD genes, and with a very ancient transspecific polymorphism. So this means that there have been two very ancient evolutionary strata around each of the PR and HD gene. And then independently, these PIR and HD loci became linked together. Each time you have the red arrow, and then it extended in some species. You can see that the orange stratum is only shared by these species, so this means it arose here in the phylogeny, so much later than the linkage between HD and PR loci, and much later than the purple and blue strata. And here in this group of species, you have the very recent red and green strata with very low level of divergence, and that are present only here in this species. So the level of divergence is not that high, but actually, so this is the ancestral gene order, right? But now this red gene, they are right in the middle here of the mating type chromosome, so meaning they definitely don't recombine, right? They are within this chaos of rearrangement. And between the HD and PR genes that are completely linked together. So definitely this region do not recombine, it has been incorporated, but very recently within the mating type chromosome. And so we also check with transspecific polymorphism, as I explained previously, when recombination suppression occurred, and we also support this stepwise extension of recombination suppression. And so for this microbiome ligninase dioiche, here is the scenario we have reconstructed. So you had initially the HD and PR chromosome, and then recombination suppression extended. So because in all the basidomycete fungi, only the PR genes themselves do not recombine. But here in this microbiome fungi, it extended to genes that have nothing to do with mating type determinism, but the recombination suppression extended around each of these loci. And then later, again, another evolutionary stratum further from the PR locus, and then a chromosomal rearrangement that linked together the HD and PR loci, and then a rearrangement, and then further extension of recombination suppression, and then again. So you had really a stepwise extension of recombination suppression, just like on sex chromosome. Another example on this species, on Silene paradoxa, you can see here the DS plot, and actually, so you can see that the whole mating type chromosome is non-recombining. There is just one small piece here with 0 DS, and that is still recombining. And you can see a very recent but very large evolutionary stratum here, and another one here, and probably several different evolutionary strata here, again supported by their presence-absence and by the transspecific polymorphism. And the scenario we reconstructed for this species was so the blue and purple strata and orange stratum, and then the HD and PR loci became linked together by suppressed recombination. And then there have been extension of recombination suppression later in the light bronze stratum. And then actually there have been introgression from another species that restored recombination. But then again, there was suppressed recombination, and then further extension of recombination suppression in this young pink and white evolutionary strata. So again and again in this microbiome fungi, there's linkage of the mating type loci and extension in several steps of recombination suppression. And so the question is why? So there are no difference between mating types, where you could imagine that the alleles at the gene could make a beta A1 or beta A2. They are just haploid cell mating and just within a tetrad. And all these evolutionary strata, because there have been different rearrangements between the species, they trap different gene sets. So it's not the same genes that are non-recombining in the different microbiome fungi. So it doesn't go, it doesn't support either antagonistic selection model. So here you have the different evolutionary strata that we name after color, and that you can see here in what species they are and when they evolved. So repeatedly you had stepwise extension of recombination suppression and trapping different gene sets. And also in other fungi, we found evidence of stepwise extension of recombination suppression. We found that in the bottom mushroom, in some coprofile fungi that we found in Wabid dung, in the oomycete also, the Plasmopara viticola is oomycete, so not a fungus, it's closer to brown algae. In Neurospora tetrasperma too, and so in both ascomycete and basidiomycete fungi, we found several cases with stepwise extension of recombination suppression. So just to show you the strata in the Neurospora tetrasperma complex. So again, it's a complex of several species. And so you can see again the recombining region with flat, zero divergence between the two mating type chromosome, and then recombination suppression. And you can see that in some lineage, you have here a high divergence and here no divergence. And when you have no divergence, the mating type chromosome is still collinear. While when you have high divergence, there have been rearrangements. So again, there have been stepwise extension of recombination suppression in this fungi. And here the very recent paper in this coprofile fungi, where you can see again the divergence between the plus and minus alleles along the chromosome, and because they are selfing, you have zero divergence when it's recombining, and around the mating type locus, you have a high divergence region, so recombination suppression, and we check that with segregation analysis. And you can see you have 3 evolutionary strata that you can see with significant different levels of differentiation. And so the first one just around the mating type locus, and then at the two sides of them. So to sum up, there is stepwise extension of recombination suppression in several fungi, despite the lack of sexual antagonism. So these mating types are not associated with male or female function. You have really no different phenotypes between the mating types, and actually they mate within the tetrad, so without any haploid phase where they could express different traits between cells of opposite mating types. So the question is why is there stepwise extension of recombination suppression? And so one interesting observation is that so this recombination suppression is present in multiple fungi, but actually overall in all fungi is quite rare, especially in ascomycetes is very rare. And the ascomycetes, most of them are haploid. And when we observe recombination suppression, it's only in the very rare fungi that are not haploid but dikaryotic. It means that each cell has 2 nuclei. So it's like functionally like if they were diploid, but the 2 nuclei do not fuse, they remain separated in the cell. So you can see here the coprofile fungi, for instance, they produce spores where the two opposite nuclei of 2 opposite mating types are compacted together. So they are self-fertile spore, and they grow by not a haploid hyphae, but a dikaryotic hyphae with the 2 mating type alleles. And so we thought that maybe the force behind the underlying this extension of recombination suppression could be the sheltering of deleterious mutation. And so we developed a model to test this idea that the recombination suppression could actually be selected for sheltering deleterious mutation and not for because of sexual antagonism. So the idea of the model is that so we know that there are many deleterious mutations segregating in genome, and we know for long that so already Ney developed some models showing that if you have by chance an inversion that carry fewer deleterious mutation than in average in the population in this region, it will have an advantage, right? It has fewer deleterious mutation, and it cannot recombine with the rest of the population because it cannot incorporate more deleterious mutation. So it has an advantage, and it should be selected for. but up to a point, because if it becomes too frequent, when it increases in frequency, it can be found as homozygous, and at homozygous, it will express this recessive deleterious mutation. And so when it's frequent enough to be frequently homozygous, it will have a disadvantage and it cannot reach fixation. Except if by chance, this inversion carries also a permanently heterozygous alleles, like the male sex-determining alleles on the Y chromosome. So if you have an inversion that traps the male sex-determining allele on the Y chromosome and fewer deleterious mutations than average in the population, and I mean, half of the inversion by chance will carry fewer deleterious mutations than average in the population, so this is not very rare, then it will have an advantage, and it can reach fixation because it will never be homozygous, right? It will never, its deleterious mutation will never be homozygous and pay the cost of the recessive deleterious mutation. And so they can be fixed in the population of the Y chromosome. And we show that indeed by simulation that it occurs. So you have here, we simulated the XY like chromosome and an autosome with so deleterious mutation segregating in the population and inversion occurring regularly. So on the autosome, the inversion can sometimes, and you have on the Y-axis, so you have the successive generations. And here on the Y-axis, you have the frequency of the inversion. And you can see that on autosomes, inversion sometimes increase a bit on frequency, but they never go at high frequency. But on the XY chromosome, actually, and especially on the Y chromosome, inversion fixed regularly and extended the region of suppressed recombination around the sex-determining gene, because so when inversion captured fewer deleterious mutations than average in the population, they were selected for, and when they trapped were associated with the male sex-determining gene, they were never homozygous, and so they could invade the population of the Y chromosome. And this occurred repeatedly, so extended, really generating this evolutionary strata on differentiation. And so this worked in the model with the XY chromosome, but it also worked with a mating type chromosome, and actually it worked with any locus that has a permanently heterozygous allele, so either the XY chromosome, the mating type chromosome, but also the supergenes like in butterflies or in ants. So one issue is our fungi. So as I told you, all the fungi where we show the recombination suppression are highly selfing. So you would say that because they are selfing, the selection should purge the deleterious mutation, so there should not be many deleterious mutations segregating in the genomes. So we did also an extension of the model where we analyzed the probability of the deleterious mutation accumulation. And indeed, when you increase selfing, the probability of deleterious mutation accumulation decreases. But actually not near the mating type locus. So because the mating type locus is always heterozygous, it protects, it shelters a bit deleterious mutation, and it allows deleterious mutation to accumulate just in the margin of the mating type chromosome. So you have here the distance to the mating type locus. So when you're closer to the mating type locus, even with selfing, you can still have deleterious mutation accumulating. And actually, especially under automixis, so intra-tetrad mating, like the microbiota fungi, but also all the fungi I mentioned, they are all automictic, so with mating within a tetrad. And so this allows deleterious mutation to segregate near the mating type loci. So this model could really work in this automictic fungi. And so we try then to see whether we could test some of the model prediction with our fungi. And so one first validation is that indeed all the species in fungi or mycetes where you have differentiated mating type chromosome with stepwise extension of recombination suppression, it's only in eukaryotic fungi, not in haploid fungi. So this is very consistent with the model where the deleterious mutation obviously can only work when you are diploid or dikaryotic. So you have here a phylogeny, for instance, of this. So this the dung of several herbivores where we collect these fungi. And so you have here a tree with the many haploid. So you have many haploid species, so producing a spore of either plus or minus mating types. But you have some groups of species where they produce dikaryotic spore with the nuclei of the two mating type, and several times independently. When you have evolution of this dikaryotic lifestyle, you have also evolution of recombination suppression, and independently. So you have independent association of these two traits, which really is consistent with this model of selection of for sheltering deleterious mutation of recombination suppression. And another interesting, so we looked at the deleterious mutation in terms of gene prediction, so genes that are the stop codon that are obviously disrupted. So in this dung fungus here, where we identified 3 evolutionary strata, and you have here with the black traits the deleterious mutation, so the disrupted gene we identify in one mating type and not the other, and they were precisely at the margin of the evolutionary strata, just like if they really were selected for sheltering this deleterious mutation. And then, so we also studied the deleterious mutation accumulation following recombination suppression. So because we have this multiple fungi with independent evolution of recombination suppression, so both events of linkage of HD and PR loci, but also multiple independent strata. So we had actually 21 independent events of recombination suppression that we could date in million years. And so we calculated, we estimated some different measure of deleterious mutation accumulation in these different strata of different ages to see the dynamics of deleterious mutation accumulation with time. And so first we look at the frequency of optimal codons. So you know that some codons are better, are more efficient, even if the genetic code is redundant. Some codons are more efficient because of maybe the tRNA are more abundant or any other reason. And so we look at the frequency of this optimal codon with time since recombination suppression, and you can see that the frequency of optimal codon decreases, but smoothly with time, and then quite seems to reach a plateau that is actually above random expectation. So it seems that selection is still a bit efficient to keep some frequency optimal codon above neutral expectation, but still it decreases this time. We also look at the accumulation of non-synonymous substitution. So we looked at the difference in non-synonymous substitution with time cis recombination suppression between the two mating type chromosomes. And it seems like they really accumulate linearly with time. And so when you compute the ratio of the non-synonymous substitution over synonymous substitution, so which is a proxy for the strength of selection, it looks like it's quite flat, meaning that you have like a steady strength of purifying selection that is neither 1 or 0, but is intermediate between purifying selection and neutral evolution as expected. So selection is less efficient than when there is recombination, but there is still some selection, again, non-synonymous substitution. And we also looked at the accumulation of transposable elements with time. So we looked at the percentage of base pair occupied by transposable elements. So they initially increased very rapidly, but then reach a plateau like in 45–50% of a base pair occupied, and then didn't accumulate further. So maybe the host genome could manage to control them at some point. So yeah, in conclusion, we developed some like general theory that can maybe explain a stepwise extension of recombination suppression on sex chromosome, mating that chromosome, and other supergenes based on the sheltering of the deleterious alleles. And with that, I'd like to thank all the students, collaborators, postdocs, technicians, many people over the years that were very helpful and amazing to work with, and all the funders, and you for your attention.